of information that we use to try to interpret what's going on the seafloor. And so when we're doing mapping, like the Nautilus does and other NOAA vessels do here on the West Coast and all over the world, it's an acoustic signal that's sent blasted down to the seafloor. And there are two kind of measures or readings that we can get from that data as it bounces back and is read again by the ship. And one is how fast it reaches the seafloor, which tells us something about depth, and then how strong the signal is. When it comes back, tells us about what the bottom hardness is like. So the stronger a return, the more likely it is to be hard habitat, and the softer the return. You can imagine, like, if you're in a soft padded room, you know, noise is absorbed so much more than you were if you were in a phone booth where everything
That's a
just
good
kind of echoes
analogy.
around.
Right, right.
And a glass case of emotion.
So the backscatter is part of that sort of like that imaging portfolio that happens during the mapping portion of the cruise.
Yeah, I'm not super familiar with the processing that it takes, because I believe we most immediately get the bathymetry data or the depth profile. And I think it takes some processing and some interpretation to figure out the backscatter, which we are working on for the Channel Islands, because we have the bathymetry data. We're like 99% mapped in the Channel Islands National Marine Sanctuary.
Which is fantastic
and now we're
considering where we were 5 years ago.
Oh, yes. I mean, the progress is astounding, and thanks to a lot to Ocean Exploration Trust, and especially their opportunistic mapping and, you know, transiting to and from areas and really targeted mapping they've been doing for us. It's taken a lot of people and a lot of resources, but we're finally pretty much almost there.
Yeah, I think it's exciting. I mean, to be able to explore this deep and really sort of see this other portion of the sanctuary that we've never really been able to ground truth before these technology tools that we have now. It's fantastic to just see the diversity here, and it adds to sort of our puts more weight on this, that we know this is truly a special place, near shore and deep sea.
Yeah, and it just helps us better understand what's going on in our sanctuary. So we're a place-based kind of management office where we research, monitor our resources, and we're also mandated by Congress to protect and conserve them. But until you have a better idea of where everything is and what you have going on within your boundaries, it's a little less straightforward how to do all those things and track the health and status of various habitats and living resources. And so this mapping information is just, it's already proven invaluable, and we are just digging into it really this year. So it'll provide a lot of information in the years to come.
Look at that.
So we're reaching a little bit steeper of a slope area here. Seeing a lot more rocks, bigger cobble.
Not good to grab
Same
bubble.
amount of sponges.
Yep.
Yeah, it looks like right now our depth is at 534 meters. And the water temp right now is just a balmy 6.1 degrees Celsius.
Pretty chilly.
It's pretty chilly. And really low oxygen saturation here too.
Oh, really? Interesting.
Yeah, it shows it at 1%.
Yeah.
some of those
I just realized also
crinoids there.
the oxygen units have been updated since yesterday's dive. They were erroneously reporting millimoles, but now they're properly reporting micromoles per liter.
Oh, good.
So the saturation is 1.1. The concentration is, 4.09 micromoles per liter.
I have a general question that I'm not entirely sure the answer of. How do corals and sponges survive areas of such low oxygen concentration?
Can we take a look at this pen? It looks to be on hard rock. Maybe my eyes are deceiving
Oh, right there.
me.
Oh, we have a sonar return on Argus too.
So Lizzie, Meredith Everett says, they can survive here because they have a low metabolism, very low metabolism. Is that?
I mean, we're not.
Sort of.
What's our bearing again?
335.
All set there, Bob.
Okay.
And they don't move.
So we shouldn't be
helps.
heading into
Exactly.
it.
It's a
but
very
whatever
low
features
energy
there is
lifestyle.
At least 20 meters tall.
Yeah, that's also a surprise. Because the slope should be on our port side.
So is this a good or a bad?
We are seeing over the top of it.
Yeah.
The ship just completed a move, so let's see.
Lizzie, some of those more cylindrical looking white sponges, do they look like the one on the lower left corner that we were sent from?
Let me pull that up again.
Abby?
I have so many tabs open. Yeah, let me see.
Talking about over here?
Is this the wall? This rocky? So depending on how wide your windows are, if we were to count them, it'd be 1, 2, the fourth picture?
Yes.
Maybe? Okay.
That's the one I was looking at. They look similar.
They look similar. Mm-hmm. Yeah, I feel like we've seen a few, so I think when we're...
Are we trying to...
Oh, and Abby joined, so Abby can tell us.
I think we're just looking at something here.
Oh, good, good.
Was there something here you wanted to zoom on, Science?
No, we can keep going.
Okay.
We're just Abby joined, and she's the one that sent the images of the sponges that they're interested in.
So can we zoom on the sponge, the white one to the right? Sorry, Julie.
That's okay.
But I think that's the one you're talking about, right?
This one?
Yes. And there's also the one to the right, and there's a little by the crab over there too. Ooh, look at that. Abby says, thanks for looking at the sponges. I think the yellow is Michael, and we have some of those. And
yeah,
a piece of the one with the white barrels. Are you seeing the chat, Lizzie?
Yeah. Yes, this one. Can we pause and take a sample here?
Sure can. Ship's holding position.
Perfect.
Zoom out now.
Good. All right, Julie.
I just was like, we started getting into the sponges. I started going back to those pictures and going, okay, I'm going to see if I can match one.
And how are we doing this one?
Do you like a slurp, Abby, or do you think it's a grab? This is a grab, she says.
How much are we grabbing?
How much would you like, Abby?
Port light on?
on it.
She's saying there's a few different species and they're impossible to tell from the images.
She would like a piece from the top with some of the body would be ideal.
Okay. You got good zooms now?
You want more?
Just didn't know that they had enough zooms.
I've got more left.
And where is this going to go, Steve?
We can go in the forward box for this.
Cool.
Okay, so we're good? Take it?
Yep, all set.
I think there's a sardine in the gripper. Yep, there it is falling.
Anchovy.
I didn't do it.
Oh,
Apparently we flew through a big cloud of them on the way down.
Okay, zoom in. Change grip to yeah, this is something amiss with this. Doesn't close at all now. 2. Yeah, that's 4. It's barely moving.
Huh.
So Abby's saying this is an unidentified, oh boy, Lysocene sponge with a clean white barrel.
Pretty rugged.
I think this one might
I was
have to
gonna
be,
say,
might have to be a
Yeah, I think.
twist.
Yeah.
we need some scissors.
Tear and
We
twist.
have scissors. That's what I'm using.
Oh,
yeah, he has coral
okay.
cutters in his jaws.
See, there's blades on that thing right there.
Oh, okay. The blade's right there.
Yeah.
A little paragorgia coral right behind it. A little bubblegum coral.
Looks like it's cutting through near the lip up there. So Abby just said they are a dense mat of spicules like fiberglass on the sponge.
Yeah, I can believe it. Oh,
It's all going to
it's
come.
loose at the base now. You don't want the whole thing?
See, we're checking. If the whole thing is
If
loose,
the whole thing, yeah.
It's loose
then
at the base.
grab it. Oh yeah, it's not even, it doesn't look like it's attached at all anymore.
No.
Mary's saying there are some little sponges in that, little shrimp in that sponge as well. Looks
Wait,
like,
where's this going?
Go in the
Forward?
forward box on the starboard side.
You want me to
Okay.
retract the camera? Or
Yeah.
just,
I think it'll fit in there. So that's B compartment.
ready for tool tray?
Also, Abby's interested in the sponge that we're seeing in the view now as we put the white barrel sponge in the box.
yeah, the one orange one that kind of looks like a heterocone. They think it's probably something different, actually. The big globules that hang off of it are much less organized looking,
Oh,
I'm noticing,
good.
than others.
I was just gonna try and
Okay, yeah.
poke that in.
That's a great sample. Abby says
Oh,
thank
Yeah, and
you.
Abby's
Sorry.
also,
Sorry.
saying
All right,
that
go ahead,
um,
roll it, Jordan.
Yeah, it's in there. Sorry.
we do try to get small pieces, but the whole sponge can actually be way more diagnostic than just a piece. So the fact that it kind of became loose and we grabbed the whole thing will be probably more helpful than just grabbing a
So
piece.
which
But we
sponge are
do
we
try
looking
to
at? The great big one or the...
sample responsibly.
This big one here, yeah. The next sponge is likely a slurp. Or so you'd like to get a little sample of this sponge too, but with the slurp.
Okay, the big orange one?
The big orange one, yes.
Yes.
Paul, you're full down on Argus cam, right?
Yeah.
From the top. So do a slurp from the top of this big orange
What was that,
sponge.
Sim? No, I'm south looking north. No, that's incorrect.
Good morning, Lizzie and Julie and team. We
Good
are.
morning, Laura.
Hi. How's it going?
We're on a sponge
Great.
hunt.
Yes,
Yeah, I think
we
if
are.
we
What
just
a
keep going the way that
beautiful
we were already going,
sponge.
we weren't on
That
like,
is
a collision
gorgeous.
course.
What is that sponge you're looking
Yeah.
at right now?
Excellent question. So typically this looks like a glass sponge known as the genus heterocone, but we actually think it might be something else. So heterocone sponges usually have these, they look pretty darn organized, the globules coming off the sides, but these are a little bit more chaotic, so we actually think this might be a different species. And so we're discussing if it's possible to take a sample just from the top lip of this sponge, but we're also considering if it might fall over, which it shouldn't unless we bump it too much. So if we just kind of grab and pull from the top, this sponge should be fine.
Okay.
Yeah.
But we can see how the pilots feel about that. And
Then a
it'll be
slurp from the top, she's saying.
Are we gonna use the slurp hose or
The slurp, yeah.
grabbing the slurp? With the
She
slurp
said
hose?
slurp from the top.
Go to the number 1 bucket.
Yeah.
I got a bonus anchovy in that.
Can you do a zoom first, please?
Is that 1 or 7? I think that's 7.
Either one. They're all empty. Whichever one you end up on.
We're on bucket 1 now.
Okay.
Yeah, zoom in out.
Thank you.
A bunch of Cheetos.
They do. Cheeto puffs. Which I think says something. I don't think I really thought about that with the typical heterocone glass sponges. And in terms of sampling this nice, lovely sponge, glass sponges can regenerate from minor damage. And Amanda Kahn did a study that looked at the regeneration of damaged sponges in glass sponge reefs. And so even though we're taking a part of this animal, the rest of it is still filter feeding and functioning because it's made up of a ton of cells that all have similar jobs. So it's not like we're removing an organ, for example, from an animal. So this should be okay if we take a piece from the top.
That's really good to know. It's hard to tell how big that sponge is. Do you have a sense of how big it is?
Zoom out.
Yeah, if and when we zoom out, you'll see 2 green dots at the center of the screen, which are lasers that are spaced 10 centimeters apart. And so if we take a picture of this, we can use the green lasers as a scale and count how many widths of those 10 centimeters. Is it tall? Is it wide? Is it deep? So it's kind of hard to do. Well, we're actually pretty darn still right now. So maybe 40 centimeters across the top.
Run a flush on it.
And
Yeah.
maybe 70 meters tall.
Good idea.
Meters, centimeters.
You got a fish in it.
That would be amazing.
Another sardine.
Sardine in the flush.
It's a really good
There's a
size,
sardine.
though.
And we have seen
Right.
a lot of these on this dive, so we are not taking the only one or taking a piece of the only one, which is also another important rule we set for ourselves for collections in the sanctuary in general,
That looks
and
good.
then especially
Right.
within a reserve.
So for viewers watching at home, if you've been watching a lot, you may have to update your browser,
Alrighty.
or sorry, clear your cache in your browser to see
zoom
the
in
green button to say that we are taking questions right now. So
Okay.
even if you see a little red dot saying we're not, we are. So go ahead and type in your question, and we will be happy to try to answer that. So someone suggests that a common name for the sponge could be packing peanut sponge.
Mm-hmm.
Circus peanut sponge.
Peanuts. Yeah, all of those.
How much are we going for here?
We've got 2 or 3 big chunks right now. Sure, you can say stop when.
Okay,
Got enough?
I'm waiting to see.
Yeah, she hasn't commented yet.
Okay, you want to, is it all in there? Turn off the suction.
Abby's saying, hold on.
Got the buckets in channel 3. Starting to drift down to the bottom now.
I think there's more even
I
at
wonder
the
what
very
Yeah,
the-
there's quite a bit more.
Coming
You can
down.
see it in the top of the screen, I think.
Yeah, you can see
couple
the
large
rough
chunks.
outline slowly. She might be switching screens to see how much is in the jar.
Steve, what sample ID is this?
This is 113. And I mean, we didn't move very much, so 112 and 113 are probably on the same marker.
Thanks. And this was a potential heterocone, or we're not sure?
Yeah, and then she's, Abby just said, great sample from the top, and can we move more slurp at the bottom of where we already slurped? Does that make sense?
Yeah,
To get a piece of one
she
of the
wants-
protections.
Yeah.
All right.
Not at the bottom of the sponge, but yeah, just a tiny bit lower than where you were slurping before.
She might have meant projections.
Yeah. Oh, instead of protections, projections, yeah.
Mm-hmm.
It's all good.
So how long does it take
You've
for
got
a sponge like this to get so big?
That is another wonderful question. And I'm not sure if many people have done much work
That's
on.
perfect.
Good?
But I do know that Abby has told me before about
Abby, is that good?
there's not a lot of data on growth and age. However, they are looking at more recently sponges growing on shipwrecks because they have a good idea of when the ship sank. And so they have a good idea of, by visiting it through time, being able to
Yes, we're
measure
good.
growth through time.
Thank you.
So I think that information is still in development and our understanding of that is still in progress. But shipwrecks are helping, so it's interesting.
Yeah, cool. Thank you.
We got an ophiuroid as part of the sample.
Yeah.
Well, nice
There's a question about how long will it take for the sponge to regenerate?
That was my question too. That's a great question. About 100. Oh, a sponge like this, it could be 100 years for it to get that size. Wow.
Wow.
Okay with the DVL reset? Great, thanks.
That's amazing. It's a long-lived sponge.
In terms of regeneration, Abby was saying Amanda Kahn did a great study looking at regeneration of damaged sponges on a glass sponge reef. But I'm not sure we have a great idea in terms of regeneration, but some glass sponges where they have done studies can grow 2 to 7 centimeters a year.
Yeah, so regeneration is much faster.
Science, are we ready to keep moving or anything else here that you'd like to take a look at?
So keep in mind you have a wall or slope, I think within 5 meters or so, I think on your backside there.
Can we take a look at this slope?
We've got a lot of classrooms watching right now, which is really cool. We've got Miss Blakemore's class, and we've got Coventry High School in Rhode Island. I'm not sure if those are the same classes, but we definitely have a lot of students online watching right now. And oh my gosh, the questions are just flying in. So maybe while you're transiting, someone wants to know who develops the mechanical grippers and controls the technology for sample acquisition and handling.
And before we get to that question, Science, do you want to continue up the slope or start heading over to waypoint X? I mean, yes.
Yeah, can we take a look at the wall that's showing up on the sonar, and then we can start heading northwest.
Okay, that sounds great.
Thanks.
You want to go up this wall or crab along it?
We can let's travel along it unless we see anything that grabs our attention to head up it.
Just a little bit more information from Abby on sponge regeneration. They cut off a part of a Afrocalistus and it regenerated within a year. So it's a similar type of a glass sponge to what we're seeing on our dive right now.
Cool.
And then pilot or nav or Steve, do you want to answer the question about who designs the technology, like the manipulators, to get the samples and handle them?
Yeah, I can take a stab at that one. So there's different, commercial and industrial companies that produce various grippers and robotic arms. So our robotic arm is manufactured by the company Kraft. Schilling is also a popular one. So some of these are off-the-shelf components. That said, there's an incredible amount of knowledge that goes into combining the different commercially available components into a fully functioning ROV. Hercules wasn't something that we just bought off the shelf. It's been, assembled with a lot of knowledge and experience from people like Bob. On top of that too, but then there's also a lot of active research in the area.
Zoom in now.
There's a lot of active research in the area of gripping and manipulation. So At some point we'll pan down and you'll see the front porch and there's these soft squishy fingers on there, which just kind of goes to show how much new and different technology is still always being developed and tested and integrated in. So we're always looking out for new methods and techniques as well.
Okay, zoom out. Look at the squishy fingers.
Yeah, so compared
Thanks
to
so
the
much.
hard metal fingers that are normally on the gripper, these are soft silicone fingers actuated by pressure.
We've got a question about, are the sponges floating around down there, or are they actually attached to something?
They're attached to the rock.
Attached. Mm-hmm.
Yes, and this area can have a lot of currents, so attachment's a good thing. And these sponge communities like to grow in areas of current because they're completely reliant on food coming to them. They're filter feeders. So they're relying on a lot of the deposits coming down from the surface and the currents and what they carry. And so we tend to find these sponge aggregations in areas where we have good rock attachments and also good current.
This is such a beautiful dive. We're seeing so much. How come we're seeing a lot more of these sponges and corals in this area than some of the muddy areas?
I think the hard substrate makes for a good attachment point for the sponges.
Yes.
Compared to the sediments. Now, there's some of you might have been noticed too. There's been that there's a red king crab that's in one of the big white sponges that we went over, and Mary Wixton says it's a Lithodes kusi or kusi, Lithodes kusi. And Abby mentions that let's see, sponge produce larvae that can swim with in quotes or crawl, but once they settle, they attach. So they got to pick a good home from the beginning.
Another factor that is particularly important for corals is that whenever you have hard substrate that juts out into the slope, for example, you create what are called topographically induced flows, which means that the water speeds up to move around certain types of topography in the slope. And so by speeding up, it can actually increase the flow of particulate matter to species that feed on that particulate matter, like corals.
Oh, there's another one right under the porch too, Bob.
Okay.
And so if you increase the flow, you increase the food, and it increases the likelihood that corals and sponges might grow larger. Oh, there's another Peristinella species.
Yeah, little coral.
There's that great shot of that red king crab, Lithodes kusi.
In the sponge.
In the sponge.
So there should be a second crab and a second sponge.
Can you zoom out, Al?
Yep, right down there.
Is there technology that the pilots use to gain a sense of depth perception when operating the arm, or does a lot of it come down to skill and experience?
Yeah, it's an experience thing because everything's kind of 2D, so you have to take in the various cameras and yeah, it's a skill you kind of develop over time.
This one.
Okay, zoom in.
Is that 2 in there?
Yeah.
Nice. Great shot.
That's beautiful.
Turn off the lasers.
It's definitely like a orange creamsicle color here, which heterocone can be kind of a bit of a more yellow pale color. Definitely orange, but I feel like this color right here is definitely more unique, and in addition to the way that the protrusions are looking, it could offer us clues that this is something different than the other heterocone glass sponges we've been seeing in other areas.
Number of sponges we've collected on this expedition, when they come up, they sometimes change color or they'll lose color, and especially when we place them in preservative, like alcohol, it actually drains out the color almost entirely, and that this is one of those sponges that it happens to. So I'm not sure what the orange color is, but they generally turn much more pale in alcohol.
When you take samples of those, oh, go ahead.
I was just saying, another reason these images are so valuable, you might be able to bring up samples in a trawl net or something, but this gives us an idea of what they look like in situ in the field.
And when you do collect those sponges, what are you doing with them? What's the research that's going on when you bring them back up to the surface?
Lasers back. Yep.
So most of these that we're collecting, our primary goal is to do more genetic work. Sponges are a relatively understudied group, especially relative to corals, and particularly in the deep sea. There's an incredible amount of variation. Even just in this one shot, I see 1, 2, 3, 4, 5, 6, 7 different kind of morphologies, different shapes and sizes and colors, and sponges can really vary their body plans depending on the environment they're in. And so one species in one high flow environment might actually look quite different than a sponge in a low flow environment. And so because they can be so, just the structure of them can be so variable. And so collecting them is really the only way to, a small piece is good enough to extract the DNA and then build, our taxonomic understanding of what is what and what's related to what. So then once we have a piece of one, we can hopefully go back through this video, and once that's identified, we can go back and enumerate the species that we've seen based on that DNA. So we'll be doing mostly genetic work on these sponges and corals.
Very cool. We have a very enthusiastic. Go ahead.
Mary Wixon was coming on that back to that sponge with the 2 king crabs in it. They're part of Paralomus virilii. Paralomus virilii. And the color for those are not as bright red, but the legs are somewhat flat. And the fact that there were more than one crab there, they could be courting. So just some more fun facts about some of the crabs we're seeing associated with these sponges. Yeah.
Oh, and a tiny paragorgia on the left, that little stick. Starting to see some more corals here.
And Abby mentions that the sponge samples will go to the Museum of Comparative Zoology to do some taxonomic identification, genetics, and the U.S. Geological Survey folks are planning to do isotope analysis on those samples to see what they're feeding on.
Okay.
Great. We have so many enthusiastic questions here, and one related from Miss Blakemore's class wondering why the crabs are in the sponges.
We zoom in on this one too, Al?
I think it's because it provides them habitat. They're not quite out in the open. They're in a slightly more enclosed area. We see crabs in sponges quite frequently, and then we see other species of crabs and crustaceans on corals as well. And so I think it's just a place that provides them
zoom out.
Shelter and potentially food. They can pick things off, or they can scavenge in and around them. But I think mostly shelter.
So science.
I was wondering if they would also be opportunistic feeders and pick off some of the shrimp and things that congregate inside the sponge.
That's a different sponge with another 2 crabs, right?
This is more pinkish.
It looks like it has a secondary branch coming off.
Yeah.
Okay, zoom in now.
Oh,
Were you saying something, Sam?
I was just going to ask you, we do need to put in a ship move soon. So do we want to continue heading north or start our northwest track to waypoint X?
Yeah, I'm interested in moving northwest towards, it looks like a little flat area. So if we can kind of transit across that on our way to X, that would be great.
Let me just drop a target in there to make sure we're talking about the same area.
Yes, yeah, that's it, right where your mouse is.
Perfect. Okay.
So Abby commented that it looks like this sponge has been damaged and it's growing back in this odd morphology. Oh, and you can see there's probably at least 3 of those Paralomus virilii. I'm going to get this right. Those are members of the king crab family. So look at the legs. They should be somewhat flat. They look like they're kind of flat. I see 1, 2, 3.
Is that another little crab on the outside there?
Underneath.
Right on the lip?
Yeah. It's an exclusive club. He's not allowed in.
There's a question about it's a really interesting question. If you took a sample from a sponge and later put it back, would the sample reattach to the original piece over time? And would genetically distinct sponges of the same species fuse together if they grew close to each other? I have a 2-part question there.
Well, if we did put a piece of the coral back, sponge back, my goodness, sorry, I'm reading too many things in the science chat. If we did put a piece of the sponge back, I'm not sure what would keep it there. So if there's currents in the deep sea, I think logistically, I'm not sure how that would work, but if we were in a more controlled environment, say the lab,
Okay.
I think I would be doubtful, but do you have any thoughts, Julie or Steve?
I think it would be very hard to have it reattach in situ in the environment, just because of all the other variables at play. I think it could get swept away pretty easily. But Abby just commented that some sponges can reproduce asexually from fragments and reattach. Some mangrove sponges are known to do this. And then just real quick, Mary, I know we just left that sponge with the king crabs, but they do use the sponges as shelter, and they might also collect some food particles, and they can take refuge from octopus that might eat them. And those crabs usually eat hard-shelled smaller organisms like crabs and hermit crabs. Clams and hermit crabs. There we go.
Someone's asking if you can compare the size of the crabs and the sponges to a common object that you might see, like are they bigger than a lemon or smaller than a lemon, or something so that people can get a sense of how big those are.
Well, we had the 10-centimeter lasers in there. For those of you that are viewing, those lasers can really help you get a sense of scale on what we're watching on the screen. So look at those and know that they're 10 centimeters apart, and then you can sort of, figure out how many of those it would take to get across the width of the sponge or a crab. You could measure the carapace, or go from the leg to leg tip to leg tip to just sort of get a sense for the what we're looking at, what the size is.
And 10 centimeters is roughly about 4 inches.
Thank you for that.
So that's about a lemon.
There you go. Bringing it all back together.
It's a big lemon.
There are some questions about if you ever
like
use
VR.
Oh yeah. Yeah, I gotta fix that. That's right around the weight.
Do you ever use VR headsets? Sure.
Hold position.
So the problem with a VR headset when you're on a moving ship is that the image doesn't match the motion of the ship, and you last about 2 minutes before you get really seasick. Yeah. So that doesn't work out as well as you would hope. So that looks like maybe you ought to do a spin.
So science, we're just getting our tether sorted out. You can
I was
see on
actually
Argus
just
channel that
thinking
we've got a
if
little
you
loop.
just spin 360,
Thanks, Sam.
you're at negative half a wrap, and I think we could just go to positive half a wrap.
Okay, that's up at the weight.
Oh yeah. All right, then
Well, I mean, we could try it. We're gonna have to stretch it out to get.
I'll come up a little
I
bit
don't
either
know.
way. Yeah, I'll do a spin
I think,
first.
yeah. I don't want to get too far back here. Can't see it.
Yeah.
Yeah, just do it, and we'll see what happens.
Yep. I'm gonna go counterclockwise.
Yep.
There's a question about the iron plates on Hercules and what those are used for.
Oh, we use those for ballast. So if we end up doing, say, geology dive and we're picking up rocks, we have to compensate for those rocks we pick up. So we can get rid of the iron plates. They're sort of biodegradable because they're just unfinished iron, so they rust away pretty quickly. And there's manila line on it, which is biodegradable. So.
Okay, we're good on the tether?
Yeah,
Perfect.
looks good.
Okay.
Bridge, Nav?
So one of the things we have to do is make sure that the tether is free from twists.
30 meters, bearing 304.
So we don't do any damage to the tether. So every once in a while, that's what we have. It's nice to have the Argus looking down on Herc. And every once in a while, we have to do some tether management.
It's like a kink in a garden
kinked.
hose.
Exactly, yeah.
Except the garden hose has fiber optics.
and 3,000 volts of, 3-phase power.
And if you don't get it sorted out, you lose video, not water.
Yeah. Bad things will happen. Abby just commented some more on that previous question about regeneration and said there were some early experiments where scientists blended up 2 different sponges, and then the cells from the different sponges separate out and formed new clumps of their tissue. So it's kind of interesting experiment.
Ship move underway.
And the ship move was 305?
304. Close enough.
Do you ever see traces of human activity on your dives? Things like fishing nets?
Funny you
Too
should mention.
A little too soon.
It's still. Last night we had to end the dive early because what was it? Was it a fishing pot line, or what? Did you guys determine what the source of the fishing debris
we didn't
was?
get the whatever the anchor was. We didn't get that up. So, but the floats and about, I don't know how many meters of line, a 3-foot pile of fishing line, like big line, like 1/2-inch diameter nylon rope, not like, you know, monofilament. It was not good.
Oh. But no, I mean, obviously we're diving again today, so no serious damage to any of the components from the.
Yeah,
the
we were
line.
fortunate there. It could have gone really bad.
Yeah.
So that's something we've
in
been talking
addition to
about with
fishing
this
line-
expedition. Oh, sorry.
Well, that's
sorry,
twice
Lindsay.
now this season that we've been caught up in fishing gear. Oh.
Yeah. Sorry, I was just to say, Julie, like in addition to fishing gear, we've also seen evidence of pollution. So we've seen glass bottles, aluminum cans, plastic bags. We've seen PVC piping and tubes and other things like that. So definitely lots of different kinds of marine debris here and many different depths and areas of the sanctuary.
And so we are on the lookout for any lost fishing lines or gear here at Footprint Reef. Historically, this has been it has been fished. It's an important habitat for rockfish, and so we have to be on the lookout for that. There's not active fishing anymore inside the Footprint Reef. It's protected as a federal marine reserve. Then that means there's no take of any kind allowed. However, on the edges of Footprint Reef, it is still fished, and so we just have to be on the lookout for any sorts of lost fishing gear. It could be monofilament line, whatever it is. We just have to be on the lookout for it and be conservative. Uh-oh,
Yep, be
lost
vigilant.
my camera feed.
So the problem with the line last night was that it was up in the water column above where our cameras are, so we couldn't see it, you know?
Mm-hmm.
So it got hung up high, so we didn't notice it until we started dragging the anchor.
So there was an anchor attached, or like a pot?
There was something. We couldn't go back
You
and
didn't
look
see
at
it.
it.
Got it.
Yeah.
Yeah. Okay.
We're going
I
304, Bob.
understand now.
Just to let you know.
Okay. I'm sort of drifting.
Yeah, that's what I figured.
About how far until we reach that target you dropped, Sam?
We've got 120 meters.
Can we start a transect real quick before we go back to questions, and then whenever we're finished, we can just head due north and hit that slopey part?
Pilots,
Zoom in now?
how are we feeling about a transect here on this slope?
I don't know. We could try it. I mean, you know.
If it's not good habitat, that's fine too.
Yeah, it's going to be more of a crab transect, so crabbing along the slope if you want to keep the slope in view. Otherwise, we'd have to, if you want to go kind of straight on, we'd have to.
Not thrilled with it. It's the fishing gear thing.
We can wait because we're going to come up to some flatter habitat. So I'll keep that thought in the back of my head.
Okay. Yeah, if you want
Looks
the
like
slope
we
in
have
view,
another.
crabbing across is not the ideal way to do it.
Okay. Okay. Thanks.
So we're seeing a Dover sole, another one of those squirrel tail sea pens.
Snailfish.
Ooh, look
Zoom
at
in
this
on
coming
him.
into view here.
Oh, nice.
Nice.
Oh, free-floating snailfish. This is an awesome fish, guys. It's worth a zoom. Look at that.
Gorgeous.
Wow.
Want to kill the lasers?
Shiny.
Yep.
Look at the colors on that. That is gorgeous.
Look at their eyes. They're so cute.
That's a
Oh,
3.5.
wow.
That's beautiful. Can you tell us more about the snailfish and what they eat, and do you have additional information on them?
Hold on.
Maybe. I'm not super familiar with them, but I can definitely, we have some guides here.
Yeah, I'm looking it up right now. So the pelvic fins in most snailfishes, I don't know if it's this one for sure, are modified into a sucking disc, with which these fishes can cling to rocks. The disc is absent in some deep sea species, so this one might not have it. They're found in cold water, and what do they feed on? So is this the blacktail snailfish? The disc is very small, and it's the gill slit's completely above the pectoral fin. It's pinkish, rear of dorsal and anal fins, and caudal fin is black. And the inside of the mouth is black, although we can't see the inside of the mouth. They're found from the Bering Sea to off of San Diego, and they like a mud bottom at about 300 to 5,250 feet, or 91 to 1,600 meters, and we are currently at a depth of 518 meters.
Wow.
Okay, moving
Great
on.
zoom.
It's beautiful.
He was really allowing us to.
Time for another ship move.
Guide.
Bridge nav.
Yes.
I think the camera's retracted. No?
Step 30 meters, bearing
That's all the way out.
304.
You want full junk zoom?
Yeah, full junk zoom.
Erica confirms it's most likely a blacktail snailfish. Caryoptractus millinurus. Thank you, Erica.
They're pretty common. We've seen probably one on almost every dive we've done so far.
Lots of questions about the fishing line, and if there was a dire situation similar to yesterday, would you need to send in another ROV to help untangle?
That would be really complicated because you get too many lines in the water, and you know, like last night, the line was over the top of Argus on our main cable, and like we could go back with Hercules and look at it, but if you start getting too close, you take the risk of the lines getting intertangled and making a bigger mess out of it. So what we ended up doing was just dragging it all up and recovered Argus onto the deck. So kept Hercules away from it. This makes the line easier to manage.
Yeah, I think there are other recovery strategies they try before they would consider sending down any additional technology.
Yeah, well, Argus has the capability of picking up pretty heavy loads, so you know it's on a steel cable, whereas Hercules is on that flexible tether, and we don't want to get things hung up on that.
Yeah.
So we can
It's a
pick
beautiful
up.
picture of.
Yeah.
Sorry, go ahead and finish your thought.
Argus can pick up 20,000 pounds, so there's that pretty much drag up any kind of fishing gear there was, you know.
Ship move underway.
Here. This is another beautiful shot of those king crabs
Oh wow!
taking
This is a
refuge in the
beautiful sponge, too.
sponge. The Paralomus virilii is the name of these crabs. I counted probably three or four. So you can see here, we've got the lasers on this sponge. Remember, they're 10 centimeters apart, or 4 inches.
Just turned them off for the beauty
now
shot.
They're off. Yeah, that's a really nice shot. And I saw, it looks like 3 or 4 crabs associated with the sponge.
At least 4.
At least 4. Thank you. And again, they use these sponges for shelter, maybe to take refuge from an octopus that might be after them. And they might collect some food particles in here if there are some. They're opportunistic. But they do mainly target clams and hermit crabs.
Got a question for Lizzie from Germany.
Okay, lasers on.
Said they've often heard you mentioning the word mosaic, and what does that mean exactly?
Yeah, absolutely. So when we're trying to gather some images that we're calling a photo mosaic or a habitat mosaic, what we're trying to do is collect images that overlap ideally by 50%. So we're taking a photo, moving only halfway over, and then taking another photo. So the center two pieces are hopefully identical, almost. And then we're doing this 360 degrees around an object, or we're covering vertically a patch and kind of mowing the lawn, so to speak, to make sure that each of the images overlap a little bit. And then we'll come over the top or do an X or some kind of more general feature over the top to connect all of those different images, because later we'll take